This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 273, recorded on February 21st, 2014. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, Vincent.
How's it going out there?
Pretty good. We have some blue sky, some clouds, 34 degrees Fahrenheit, 1 degree Celsius.
Nice. Here it is. Soup. Pure soup outside. Can't see the other side of the Hudson River. It's raining and 5 degrees Celsius. All the snow is melting.
Oh, we have so much. And walking home last night was walking through these huge puddles at every curb cut, because that's where it just all builds up. Oh, it's awful.
I spoke to Michelle yesterday, and she said that an ice storm was imminent, but I guess she didn't get it, right?
Well, it was pretty nasty stuff. Yeah, it was not truly ice that was coming down, but yeah, it was bad.
Also joining us today from North Central Florida, Rich Condit.
Howdy, Vincent. Hi, Kathy.
How are you doing there?
Good. We got 65 degrees Fahrenheit. I don't know what that is in centigrade, and I'm not going to look it up. And partly cloudy skies. We just had a magnificent rainstorm pass through.
Nice.
So had a cold front come through. Everything's good.
I was listening to a podcast the other day, and the host got on a rant about Celsius. I have to send you guys a link to this. He went on for half an hour why Celsius doesn't make any sense and Fahrenheit is the best. It was really pretty funny because it reminds us of our discussions.
Yeah.
Well, he's exactly wrong.
Yeah.
Well, he said basically 0 to 100 means nothing to people. It may mean something to scientists, but
wait a minute. It doesn't mean anything to Americans or British, but it means a lot to most of the world.
Yes, this is true. Well, I guess
so this is, I can actually get myself out of this. 18 Celsius.
I was going to say 18, yeah.
Pretty warm. Also joining us today from North Central Florida, Dickson Despommier.
Hello, Vincent. Hello, gang.
Dickson, you were very quiet.
Well, I was waiting for you to introduce me.
Now, what the heck are you doing in North Central Florida?
I'm here to give an after-dinner talk tonight.
After dinner.
This is true. I'm their after-dinner speaker.
Oh, you're their after-dinner drink, huh?
Something like that. So this is a series of lectures sponsored by the law school. Public Interest in Environmental Conference. That's what it's called. And it's all about how to posture science of the environment, namely climate change issues, for issues that relate to what does the legal world think about 2 meters sea rise, for instance, or how are they responding to crop losses due to extreme droughts and things of this sort. And it's been the discussion has been quite lively and quite interesting, really. So I'm sort of a surprise of, there is another way to farm type of thing, and that's what they'll hear about tonight. So that's why I'm here. So I'm visiting with Rich, and I'm having a great time. And I also met somebody here who teaches a course in soil sciences, and she actually requires my book, The Vertical Farm, for reading in her course. I can't believe that. And so I said to her, "Oh, so you're the one who bought it."
Well, soil science. You don't have any soil in a vertical farm.
No, she knows that. She absolutely knows that. And so she's teaching this from a perspective of ecological damage. And look what we're doing to the soil. It should be doing something for nature, but it's not. So that's why I'm here.
And you're heading back tomorrow?
Yep.
All right. Well, enjoy the rest of your stay.
So we are together, but not in the same room because
correct.
Skype does, and I wish that I could be in the same room with Dickson to practice your.
Yeah, right. Did he go away?
I don't know.
Sounds like he got cut off in mid-sentence.
It does sound that way.
Hey, Rich.
Yeah.
Oh, he's still there. Okay. All right, we have a couple of follow-ups. First is from David. Although I adhere to the distinction between symptoms, report of subjective experience, and signs observable to a clinician that you guys arrived at, it nagged at me. Too many instances of the type, quote, something is a symptom of something else, unquote, were at the edge of my awareness, in which vocabulary access has been disrupted by neurological event, especially access to ordinary language, cluttering me up a bit. This seemed to apply in the realm of things like plants and cars, unlike the precise distinction used in medicine. So I Googled define symptom. Guess what? Although a few sources lead with the precise medical usage, most didn't. Most endorsed examples of the type something is a symptom of something else. Word.com/iDictionary presented definition 1B as an evident reaction of a plant to a pathogen, following 1A giving our classic medical quibble. Other examples abound in the major dictionaries listed. If curiosity leads you to want to look, I think the etymology from the Greek supports our precise medical bias, but centuries of common use have placed symptom into English as commonly accepted and sometimes preferred to refer to an indicator of some other phenomenon where subjective report would be nonsensical.
Yeah, I agree with that.
But you didn't originally when we defined it.
No. We didn't get into the common usage of the word symptom.
Okay, but a physician would use it correctly, right?
Sure, and that's, I think, undeniable.
Okay. How's about that? All the best. Davey is in Fresno. P.S. I don't give a damn about Trader Joe's acceptance of GMO phobia. Well, I do really, sharing your views, because they have some pretty good stuff for pretty good prices. Contrary-wise, I don't give a damn about Whole Foods disliking its overpriced, irrational elitism and redundant shelf contents. They do have some good bulk grains and legumes and stuff, though, but I don't get there to harvest them. Anyway, catering to GMO phobics flies in the face of concern for hungry humanity, who will cause much more ecosystem damage if unfed than GMOs ever would, short of gray goo producers. Thanks for your digressions on the subject.
Here.
I like to hear that somebody appreciates our digressions.
Yeah, that's good. Well, I think we sort of know that, and that's why we do it in part, or that's why we don't avoid it. All right, we had a letter from Bryony. Bryony? Like Bryony Seas, who wrote back to you via TWiV, following on from last week's episode. Yes, the movement of ludioviruses from the gut of the aphid vector into the body cavity, hemocele, is receptor-mediated. Therefore, the host range of the coat protein toxin fusion is likely to be determined by the similarity of the receptor proteins between species. It is more likely that the fusion protein could enter the hemocele of insects that are related to aphids, i.e. other sap-sucking insects in the order Hemiptera, which are typically pests, than the more distantly related species such as ladybugs, which are beetles in the order Coleoptera. This hypothesis remains to be empirically tested, however.
Roger that.
That's good to know.
Hey, Dickson, are you in the order Hemiptera because you're a pest?
I'm not a sucking pest.
Oh.
I just hang around pests. Sort of like a whitefly disease.
See, I can abuse you and you don't get mad at me.
No, because I know it's so ridiculous. How could I possibly get angry at such a stupid statement?
Bryony, what kind of a name is that, Kathy? Do you know?
No, but I have a English friend who's named Bryony as well.
I like it. It's a nice name. Mm-hmm. Bryony Bonning, Iowa State University. And then we have one from Yegor, which I guess is Yegor, Y-E-G-O-R. He's written before. Yeah. Dear TWiVsters, you had a listener question in the latest episode about the difference between flu and cold. About 4 years ago, I got interested in the etiology of flu-like symptoms and tried to find a good study on this topic. Most of the stuff I found was very old, from the 60s or 70s. Hey, I take offense to that. I'm from the 50s.
I'm from the 40s.
Wow. After multiple fruitless searches at NCBI, I eventually found the best paper by consulting Wikipedia. The study, and then he gives a link to a study, Viruses and Bacteria in the Etiology of the Common Cold, from 1998. The study was done in 1994-95 in Finland and found that the vast majority of colds is caused by rhinoviruses, 64%, with coronaviruses 10% and influenza 7% being distant second and third. The remaining 12% was split between bacterial infections, parainfluenza, RSV, respiratory syncytial virus, enterovirus, and adenoviruses. Back then, they had to isolate the viruses to establish the etiology, so the relative efficiency of isolation is an unknown factor that we can't take into account when looking at these numbers, but they are still informative. These days, one could probably just pyrosequence the samples. I wonder if anyone has done a similar study since then. By the way, notice the 4% of colds caused by bacterial infection. That's a good number to cite to anyone who wants to take antibiotics when they have a cold.
Right, exactly.
This is a really cool paper. I like it.
The only danger here, I would say, in the future is that if people start refusing the whooping cough vaccine, you could see a rise in bacterial-induced infections that are commonly called colds, but they would lead to something quite serious.
Dickson, why are you speaking so softly?
Because I'm getting over a cold.
Oh, was it a rhinovirus cold?
I hope it was, but it was a protracted infection, so I don't know. We thought of culturing it, but we decided not to do that, didn't we?
I have a question for all of you. So, if nowadays you say you use PCR, you wouldn't have to pyrosequence, I guess. Mm-hmm. If you use PCR and you found sequences, I mean, would you be convinced that the virus was there?
Of course not. No. We've already talked about this a lot, haven't we?
So I like the old studies where they culture them.
Sure,
even though the numbers may not be
exactly
completely right, they tell you that the virus is replicating, right?
That's right.
All right, he continues, in the same segment, one of you said that most vaccines prevent viral infection. I suspect this may not be based on solid evidence and comes from old studies done before PCR or other sensitive diagnostics. Do we really know that vaccine-based protection from polio, measles, mumps, rubella, etc. is mediated by neutralizing antibodies that absolutely prevent the new infection? I believe that in many cases the infection happens but either goes by unsymptomatically or is quickly suppressed by the immune system. I can't find the studies right now, but I think I have seen some where people tried to detect such unnoticed infections by tracking anamnestic immune responses, and indeed saw that now and then such responses are boosted in apparently healthy people. Well, I think for these vaccines, we do know that the correlate of protection is an antibody response. And for example, for polio, that was studied for many years. And in most people who had recovered from polio and were subsequently immune, the antibodies correlated with that. So if you don't know what the correlate of protection is, you really can't make a vaccine. That's one of the problems with the HIV vaccine trials.
Yeah, my sense from this was that one of the points he's making here is that the vaccines don't necessarily completely prevent the virus from infecting you.
Okay, for infection, not disease. Yes. Okay, fine.
But they prevent disease. Okay, and I can't quote any studies, but I'm on board with that. That makes sense to me.
That could be.
That the virus gets in, maybe replicates a little bit, but your immune system's got a head start, so it squashes it before there's any trouble.
And speaking of mumps, there's a current outbreak of mumps at Fordham University in New York City.
Maybe that's what you have, Dickson.
No,
since you teach there, right?
No, this is at the Uptown campus in the Bronx anyway, but
so
I don't have any submandibular glandular swellings.
Okay. All right, so I misinterpreted his point. So yes, it could be that viruses, even in an immune person, viruses get in and replicate a bit.
Yeah, to me, this is one of the issues with in fact, with making an HIV vaccine, is that once HIV gets integrated and starts going, so if there's anything going, you're stuck with it.
Yeah.
Okay.
Yeah.
And so a good vaccine, it would seem to me, at least logically, would have to actually prevent infection. And my guess is that the number of vaccines that actually do that are pretty few. But maybe we'll get some more feedback on this.
I don't know how you'd study it because...
No, it's hard. Yeah, how do you study that?
You just have to randomly sample people, and that would be just not very good.
Well, there may be some sort of mouse model that you could do it in, but, By the way, the Bryony is a girl's name that is of Greek origin and means climbing plant.
Nice.
I like
and there are some actually Bryonies native to Europe that are specific. Large leaves and small flowers. It's a particular.
Well, plant. That's why she works on these viruses, right? Perfect sense. Now, what does Vincent mean? Animal virus?
No, abuser of Dickson.
I was going to say exactly that.
Now you're getting sensitive, right?
A little bit. Well, I'm exhibiting an anamnestic response. How's that?
Right.
By the way, is his volumes okay? I can go in there and turn him up if you want.
That's all right. I'll do it in post production. You just want to give him hell. You could do that over Skype.
I'll try to raise my voice. Okay, I'll try to raise my voice.
That's all right.
Hey, Dickson, how do you like the view from that office? Nice,
It's fabulous. No, it's fabulous.
What are you looking
Wish
at?
we had some sunshine shine.
I'm looking at part of the medical center, I presume. I'm looking at a bell tower to my left. I'm looking at partially blue sky, and a brand new building up in front of me, the Evelyn F. R. William L. McKnight something Institute.
Brain Institute.
It's the Brain Institute. Wow.
And it's got a really cool sculpture on the front. It does. If there were sunshine. Right. Each of those triangles refracts different colors depending on the angle of the sun and the angle of them, and it really shows up very cool.
It's
anyway. Okay.
Had you met Dickson before, Rich?
Me? Oh yeah, we did.
Yes. Come on, you were in.
We
were together in your office.
Together in the TWiV
in my office.
studio.
Yeah, I forgot that. Yeah.
Yeah.
Okay, so it's nothing new, right, Dickson?
Did you see I posted a picture on Facebook of our dinner at Satchel's last night.
I didn't see it.
You need to get in there.
All right, I'll check it out. I'll like it later.
Yeah.
All right, we have a paper to do today that's from Cell Host and Microbe. It is called Interferon Lambda Alleles Predict Innate Antiviral Immune Responses and Hepatitis C Virus Permissiveness. The authors are Sheahan, Shea Stadium, Sheahan, Imanaka, Marukian, Dorner, Liu, Ploss, and Rice, Charles Rice Laboratory at Rockefeller, also Imperial College of London. Well, the Dorner is now at the Imperial College of London, and Ploss is now at Princeton. And I know this because we tried to recruit them, and they went off somewhere else.
I'll be darned.
They both gave seminars. They both came back. In fact, we failed at recruiting a virologist. The third person we offered a job to said no. So I'm here all by myself.
Just bouncing off walls.
Vincent.
That's why I'm TWiVing, because I got these kinds of discussions, and that's all I need.
Vincent's TWiVing because he's taken what they're given. How's that?
Hey, I thought you were going to rhyme. All right, so this is an article about hepatitis C virus and what makes it. So hep C infection, as you know, or as we have talked about, many people can clear the infection, but others do not. Something like 70% of people develop a chronic infection.
Yeah, it's actually a really high number.
Yes, and then
the majority develop a chronic infection.
And that's bad because you can get fibrosis of the liver, cirrhosis, and ultimately cancer, liver cancer. And so this is one of the reasons why it's a big deal. And why this happens is not clear. Why do some people clear it and others do not is an ongoing issue. It's a really hard question to study in people. First of all, it's hard to do because you have to take a biopsy of the liver and you can't get all the samples you want. And so people have tried other things like chimpanzees, and of course you can't use chimps anymore in the U.S. for this sort of research, so that's finished. They've tried primary human hepatocytes, so you get fetal liver cells and put them in culture and infect them with virus, but these aren't infected very efficiently. There's 2 to 30% of the cells end up getting infected. It's hard to detect infection, so there are issues. And
yeah, it took me a little while to catch on to we're getting to exactly what they did, but part of the necessity here is this low percentage of cells infected, because for some of the things that they're doing here, if you had a virus that infected efficiently, you'd just do a high multiplicity infection,
right,
and harvest that. All the cells are infected, so that whatever the average is probably representative of what happens in individual cells. But if you only get a fraction of the cells infected,
yes,
then when you try and do a measurement, you're getting an average of uninfected and infected cells, and it's not an accurate picture of what goes on in the infected cells.
Right, and even in these cultures, they don't get infected well, and they say it's really hard to find the infected cells because the levels of replication are low. So even if you do immunofluorescence, it's hard to see. So they have two approaches, really cool technical approaches in this paper to solve this issue with primary human hepatocyte cultures. That's what all the work is done with here. The first is called, and I heard Charlie talk about this a couple of years ago, it's called HCV infection-dependent fluorescent relocalization. All right? So basically what they did here is they took a mitochondrial protein, Visa, MAVS, goes by a number of names, actually an intermediate in innate signaling, and they engineered a version linked to fused to GFP, green fluorescent protein, or some fluorescent protein. And when these cells are infected with HCV, its HCV protease is known to cleave this protein MAVS. When it's cleaved, it then goes into the nucleus because there's a nuclear localization signal in the fusion protein. And then in the nucleus, it's very clear that you can see the cell is infected because it's all concentrated in a small area. A lot easier to see than in the diffuse cytoplasm. So it's a great readout for saying this cell is infected.
And that fluorescent molecule, the GFP MAVS, is delivered to the cells in a lentivirus vector.
Correct.
Right?
Right. You put it into the lenti, yeah.
And in this, they make the hepatocyte cultures, and then do they infect with HCV first and then the lentivirus, or the other way around, or simultaneously? First. Do you know?
Lenti first.
First,
and then HCV.
So they establish that MAVS thing, and then come in with HCV. Do you know? I'm getting at an issue here, but do you know? Is there a time lag, a significant time lag between those two? Do you know? I wasn't able to figure some of this stuff out.
Actually, I had pulled out the original paper and I saw it, but I don't remember how long it is.
Okay, fine.
So this is a little strange because I looked up some paper, and what I sketched for myself was an NS34A construct fused to GFP, or NS34A with nuclear localization sequence fused to RFP, not the MAVS that you're talking about.
Yeah, so there are a couple of versions of this by different labs. And
this was one, I thought it was the one from the Rice lab, and I actually thought it was the one that was described for this paper, but I could be wrong. Okay.
Because I remember when he talked about it, it was, the point is it's going from the mitochondria to the nucleus. But
right.
It could be so. In either case, the idea is that infection causes the protein to go in the nucleus where you can easily score it as infected. And that's a big improvement.
And that mobilization results from the HCV gene expression because it's a protease that cleaves that thing.
Correct.
That's for sure.
Correct.
Right. So they referenced 3 papers for this HDFR, and I looked at one of them.
So let's see. I looked at which one did I look at?
I think
no, I didn't look at this Nature Biotechnology.
That's the one I looked at.
I looked at a different one.
They're all from the Rice lab, I think.
There's this Horner and Gale regulation of, I don't remember. But the one I saw had a MAVS in it anyway.
Yep.
So it could be not the one they use, but they actually don't say in this paper.
Right.
Well, they say a mitochondrial localized RFP, and that would be consistent with MAVS. MAVS also has about 3 or 4 other names. Yeah. I looked at one of these papers as well, so that gets confusing.
IPS1, yeah. Visa. Visa. Okay, so that's one technology that they use. The other one is laser capture microdissection. This is so amazing.
Yeah.
So they have their hepatocytes on a slide, and they can look at them under fluorescence light, UV light, and see which ones are infected with HCV, and then they can cut them out with a laser.
Mm-hmm.
And
they're cutting out individual cells.
Individual cells. I have pictures of this here. You can see where the cells were and then where they weren't anymore. So I looked this up, and there are several ways you can do this. You can cut the cells and actually pick them off the slide. But in this paper, they cut around the cells, and then they put buffer that would lyse the cell and pulled the buffer off directly, which is amazing. So you can get individual either infected or uninfected cells from these liver cultures.
And you can get the cells from immediately around an infected cell,
right.
which is especially nice.
Right. See what's going on in them, because with innate immunity, things are diffusing, right, as well as virus. So pretty neat combination. So you can look at single cell infection, basically, in these hepatocyte cultures where, a few percent of the cells are infected. You could go after those and say, hey, what's going on here?
So the amazing thing to me is that they pick out these cells. As I understand it, every sample is actually a pool of 20 cells. Is that right? For the microarray analysis?
That's correct.
And so they'll take 20 cells and make RNA from that, make cDNA from that, and hybridize that to a microarray, right?
That's correct. That's correct.
Amazing.
That's where a whole genome microarray, so I guess they're sampling.
And for confirmation, they'll take another 10 cells and do qPCR on it.
For virus,
okay, so they have two independent assays.
So the qPCRs, are you talking about for virus or to confirm the gene?
They do a little of both, don't they?
Yeah, they do look at virus as well, yeah.
Right.
So basically they validate this system. So these are fetal liver cultures. So they're from young fetuses, and there are quite a few different ones of different backgrounds, which we'll get to a bit later. They put them in culture and they infect them with hep C, and then they laser dissect the infected and neighboring cells. And
so they can compare infected cells and adjacent uninfected cells, as Kathy said. They can compare that to mock infection.
That's right.
And here's where I have a question, because not only are they infecting these with hep C, but they're throwing in this pseudotyped lentivirus.
That's right.
And I wonder about, if I were a cell, I wouldn't like that, okay? The pseudotype, I'm basically getting infected. It's an innocuous genome, but
sure,
nevertheless, I might react to that. And I didn't see anywhere in here where they were doing some sort of a control to look at what effect the lentivirus itself might have on this whole system. Did I miss anything, or am I being too...
No, I think it's a good point, because the cell is going to make a response to the lentivector, right?
Yeah, right.
So there's a fourth group that should be added to this, then, you're saying?
That's somewhere along the line I'd want to do that control, just to find out what effect
I
that
didn't
had.
notice it. I didn't
see it
at all in my reading of this, but that's clearly an important point. And if any of the authors are listening, or Matt Evans, let us know.
They do the HCV infection 48 hours after the transduction.
Yeah.
Okay.
So
it's enough time to get a response of some sort. But if I remember, so the mock infected, or do they just not get anything, or
they are not clear on that? Okay, so I don't know if that's really don't get anything, or if they get the lenti.
Because that would be your experiment, the mock just lenti, right?
I would, if it were me, I would even want to do
both. Yeah.
The
uninfected,
no treatment, and lenti alone.
Yes.
And see what happens.
Yeah. So that's a good point. Yeah. So they basically show that this system works, and the neat thing is that you can pick up a infected cell, and it will not have contaminated the neighboring cell. It's with the transcriptome, the RNA, basically the microarray analysis. So the microarrays, they have oligonucleotides representing many, many genes. I think probably 20,000 if it's whole genome. And then they extract RNA from these single cells. They make cDNA, probably amplify it, then they hybridize it to the microarray. So that's what they call transcriptome analysis. They don't contaminate, so the neighboring cells are not contaminated with the ones that they cut out, which is pretty neat.
Yeah, and I think that one of the best validations of that is that they can have pretty robust viral genome signals in an infected cell, and an adjacent cell's got nothing.
That's right. That's right. So the virus production, let's see here. Where does it say that? Infectious. So they actually measured infectious virus released into the culture medium. And virus production goes down 1 and 3 days after infection. Some donors secreted high levels of virus as late as 7 days post-infection. So it's basically somewhere between 1 and 7 days that you're getting virus production. But there is a decrease in these cultures, and it varies from donor to donor, that is, the donor of the liver cells.
I wonder what it is that limits the infection frequency. Can you not get a high enough titer of virus to infect them, or are there some cells that are resistant to infection, or what?
It's a good question. I think, I mean, I don't know what kind of titers they have, so maybe that's limiting it, but maybe I just don't know. Yeah.
So what's typical for most viral infections that are competent?
Well,
100%
if you're working on a really
or
cool
30%
virus,
or 80%?
Yeah.
If you're working on a really cool virus like Pics, okay, you can, easily grow a virus stock that's got 10 to the 9th infectious units per mil, and then you can choose your multiplicity of infection. And if you go to a multiplicity of infection it follows exactly Poisson distribution, anything over 3 up to 5 or 10, you infect all the cells, period. Okay?
Right.
And if you go down to a low multiplicity, you can infect a fraction of the cells. So,
but there are some
systems like this where for some reason or another, you can't get all the cells infected. And I don't know if it's because you can't make a concentrated enough virus stock, or whether some fraction of the cells are actually resistant to the infection. I don't know.
Or the viruses are incomplete.
So that would be like not having a concentrated enough stock of infectious virus.
Correct.
Depending on how you assay infectious. As I recall, they make these stocks they've got an infectious clone that they transfect into cells and then harvest the supernatant off of that.
Right.
Right.
Mm-hmm. All right, let's go through some of these experiments. So
even growing HCV and maintaining a stock is no small task. It's all very difficult.
So they made 188 cDNA libraries from these laser capture samples. They have mock HCV infected and adjacent cells. So they did microarray on all of this. And then they pooled, for this analysis, they pooled all the donors at each time, okay, to see the overall responses. And they say the number of genes regulated more than twofold over mock in infected and adjacent cells, increased from 1 to 3 days post-infection, and then decreased by 7 days. So, Rich, if they're comparing the HCV infected with the adjacent cells would have the lenti vector in them, right?
Yes.
They should.
Yes.
So maybe that's your control. Everybody's got the lenti, and then you're looking at the increase of the HCV infected over the adjacent cells.
I'd like to know whether the mock infection must have the lenti in it. That's the way you'd do it, wouldn't it be?
Yeah, I would.
Yeah.
It doesn't say in the methods?
I didn't catch it. I could look.
I didn't catch it. I was kind of looking too.
Over half of the genes regulated on day 1 in the infected and adjacent cells were associated with innate antiviral immunity, which is, I guess, what you would expect, right?
That's reassuring.
Mm-hmm.
And then they single out type 3 interferon, or also known as Lambda Interferon. Its induction was observed only in HCV-infected cells. And if they add a viral inhibitor of the replicase to the cultures, they do not see interferon lambda production. So
that's a great control. I like that.
Yeah, you got to have replication. So they say that these data unexpectedly indicate that an antiviral program dominates the response at very early times after infection. And I think it's unexpected because over the years, many people have shown that hepatitis C virus has many ways of interfering with innate responses. So I guess they're surprised that there's such a strong response program here. So the next thing they do is to say, so what are these genes that are...
Wait,
and
can I ask
the
a
cool
question
thing
right
here
now?
Yeah,
is the adjacent cells fire too?
The adjacent cells fire.
Okay.
Yes.
So that implies that they know that the guy next to him is infected.
Correct.
All right? And that is affirmation of what interferon is actually supposed to do. because the infected cells send off interferon, which tweaks the neighboring cells to an interferon response so that they are in a defensive position should the infection spread their way.
Yes, so this increase of genes occurs in the infected and the adjacent cells, correct?
So you're using primary fetal hepatocytes.
Yes.
What would happen if you were to use adult hepatocytes?
I don't know. I think it's harder to get enough of them to do this sort of stuff.
I understand that, but maybe it's a characteristic. Maybe this robust primary innate immune response is a characteristic of fetal tissues.
Yeah, because that's all they have, right?
Exactly.
Actually, I would think that, the paper we did last year worked on traffic accident victims. I would think that getting adult hepatocytes wouldn't necessarily be all that difficult.
Yeah, but maybe hepatitis C free hepatocytes from adults might be difficult.
Well, I wouldn't necessarily think that would be that difficult. Could be that the culturing them is different. It could be that they're hard to infect.
Yeah.
So, you might be looking for your watch under the wrong street lamp.
Ooh, what is that? How's that go again?
The streetlight effect.
Streetlight effect, right. Yeah, I like that. Why are you looking over here? Because the best light is here.
Right.
Exactly.
It's not where I drop my key.
Right.
That's the deal.
I like that.
Well, this whole thing is very difficult, as we've already said.
Understood.
You do the best you can.
To begin with.
Yeah. Yep. That's a good question, and perhaps someone will answer it for us.
I think we're going to get mail.
Good.
Which will be good.
All right, what genes are turned on here? They say in the HCV-infected cells, they can see 3 distinct temporal phases. In the first, you get induction of genes related to the innate immune response, along with the effectors of the interferon response, like the ISGs and also chemokines. Then you have a second phase where the inducers of innate immunity were low, and you see the effectors, like the ISGs.
Interferon-stimulated genes.
Interferon-stimulated genes, of which we've talked about here, like IFITs. The IFITs we've talked about before. Genes related to antigen presentation are starting to go up, the MHC molecules. And the third phase is marked by antiviral genes that are upregulated at all time points. And these look to me like some interferon-stimulated genes. Adjacent cells regulated fewer, about half of the number of antiviral genes. So even though, Rich, they're inducing, that's not as many, right?
Right, which makes sense.
So they conclude they have donor-independent, distinct, and dynamic antiviral responses in the infected and the adjacent cells. And they say this would have been impossible to obtain without this kind of approach. And it's true. Right?
And as a matter of fact, I think one of the really cool things about this approach, I was talking before about how, in a classical virology experiment, you would do a high multiplicity infection and assess the infected guys and compare those to a completely separate mock culture. Out of that experiment, you don't get this thing about the cells that are directly adjacent to each other, infected and uninfected. So that's a unique property of this system that's very cool. And I'm just sitting here amazed that you can get this kind of data out of picking individual cells with a laser off of this.
It's incredible.
It's beautiful.
I mean, you can make RNA and cDNA from a single cell.
These guys can. I don't know about me.
I mean, I was just thinking of this, you don't want to screw this up.
No.
You don't want to drop it. You don't want to contaminate it.
No.
Because it's kind of precious. All right, next they look at this issue. They find that even though these cultures are the same, and they put the same virus on, they get variation in the amount of HCV produced depending on where the liver cells came from, what the donor is, the person who they came from. All right, and if you look at one of these figures, you can see that there's quite a range of virus production, from like 10 to the 3 to almost 10 to the 6th TCID50s per mil per square centimeter. That's their measure. It's pretty big range. So each dot on these plots are a donor. So why is that? So they put their data, they stratified their data into two groups, which are called high donors and low donors. So now there's the highest and lowest infection frequencies based on the amount of virus produced, I presume?
Yeah, I think so.
So
it's a percentage of reporter positive is how they measure
yes,
yeah.
okay.
frequency.
Okay.
Yeah.
Yeah. So an inverse relationship is seen when comparing the two groups with respect to the number of related genes. So in other words, the high donors have low numbers of genes induced and the low donors have high numbers, correct?
Mm-hmm.
Yes.
So the low donors have high induction of antiviral genes, which makes sense. That's why they're not making a lot or they're not, they don't have as many infected foci.
Mm-hmm.
And then they can look at what responses correlate with effective control of infection, like what kinds of genes. So the low donors have a lot of inducers of the innate response, a couple of genes that are involved in induction. And they have a number of genes that are uniquely upregulated in those cells. So again, that would be, well, these are good to have induced. They help you keep the infection low. Low donors showed unique regulation of 10 antiviral genes in adjacent cells, whereas high donors uniquely regulated none. So some clues about what might be wrong with them.
Right.
So the idea is that the higher antiviral responses in adjacent cells may limit the permissiveness to infection and spread. What function is being addressed, they can look at the antiviral genes and say what's being inhibited. And they say RNA virus replication is among the top 5 most significantly affected functions associated with low donors. So if you inhibit that function, you do pretty well with controlling virus replication.
Right. So they talk about a suite of genes that can reduce RNA virus replication.
Yeah.
So,
it's really neat.
Yeah. I have one tiny complaint, and it's just that they start talking about ingenuity gene ontology analyses.
Mm-hmm.
And from what I can tell, that's a commercial product. And
it is, yeah.
It just makes it look like ingenuity gene ontology is a word, phrase, common language thing. And they should, I think, cite the company or...
Yeah, I agree. Yeah.
So
it is a software package by a company called Ingenuity, which has since been bought by Qiagen.
Right.
And it allows you to put the genes that you find in any study like this into functional groups, right? To give you some ideas about what is affected.
Right.
And that's how they can see RNA virus replication.
Right.
So,
and from the next time they mention it, when they actually call it Ingenuity Pathway Analysis, the Ingenuity is lowercase. So it would be like the trademark lawyers would get after them pretty soon.
That's true. That's right, yeah.
So.
Right, so now they look at the interferon lambda part of this story, which is in the title. And previously it's been known that polymorphisms, our old friends, the SNPs, single nucleotide polymorphisms, near or in these interferon lambda genes. I think there are 3 of them, right? Or 4. There are more than one.
Yeah, I think there's 4.
They're associated with good responses or bad responses depending on the SNP. So they want to know if they can pick this up in their data. And they actually there's a figure here that puts it beautifully. I think it's figure 4. Mm-hmm. They stratify their data. So they have donors from they have liver cells from many different donors, and they could say, what's the interferon lambda genotype at these SNP positions that have known to be important before? And they can align those with the clinical outcome of HCV. So it goes from very good outcome, which is the major allele of interferon lambda, and it goes all the way to a poor outcome, which is the minor allele. So, for example, interferon lambda 3, the major allele is CC. There's a heterozygous allele CT, which is associated with a poorer outcome, and then the minor allele TT has a crappy outcome. In other words, you don't have a good outcome of the infection. And those correlate with their data, basically. So the high and low infections correlate with these major and minor interferon lambda alleles.
Mm-hmm. So you have the major alleles, you'll have a good outcome, and you have the low infection
frequency.
Exactly. And the poor outcomes correlate with these minor alleles that have a higher infection frequency in their assays.
So if you did 23andMe, could you determine that for yourself using this data?
I don't know if 23 does the Lambda HCV SNPs. That's a good question. I'm not sure. I have to go look because I have all that data, but I haven't looked at it. They don't do everything, but they do a lot of them, and they certainly do norovirus susceptibility because I know I'm susceptible.
Don't take any long cruises.
Empirically.
You know that empirically?
Yes.
No slow boats to China.
No, I don't want to take any cruises because I don't want to be on a boat with a lot of sick people.
Yeah.
I just don't think it's worth it. But I don't want to be on a boat. I'd rather walk around Europe, Right?
Right.
Yeah.
I don't think I'm preaching to... I don't need to convert you guys, yeah. Okay, so they can look... They take their data and they stratify it by this interferon lambda genotype. And so, for example... So the numbers of regulated genes increases if you go from the major, the favorable allele to the minor one, right? So, which is the unfavorable. Numbers of significantly regulated genes increase stepwise from homozygous major favorable to homozygous minor in both HCV infected and adjacent cells. Mm-hmm. So they have more virus, so they're making more, they're upregulating more.
Right.
They have the highest absolute number of regulated antiviral genes is in the minor allele donors, whereas the majors have fewer. Interferon lambda major allele donors regulated fewer cellular pathways in addition to the antiviral response. So the major donor responses are mostly antiviral and more focused, whereas the minor allele donors, they have a response that is not necessarily just antiviral, but is broadly antimicrobial.
Okay.
Top network associated with the 32 genes unique to HCV infected cells of major donors was cell death and survival. Okay, so if you are in this major allele group, you typically upregulate genes involved in cell death and survival. 6 genes in minor allele donors were upregulated more than 5-fold over major allele donors. And cell genes related to cell death were enriched among low and major allele donors, whereas those unique to minor allele donors were largely ISGs. So basically, this shows a correlation between the interferon lambda genotype, which we've been calling minor, and major alleles. Hepatocyte permissiveness, because they can look at their infections in their cultures, and the focus of the host response. It's pretty cool.
Mm-hmm. I kept getting confused because of major and minor and up and down. This must have been very difficult to write, actually.
I suspect that they took a long time to write it.
Yeah, so
it's not easy.
Did I understand correctly? There's a I'm looking for it, but I can't find it. There's some conclusions in here about the levels of interferon stimulated genes in these different genotypes, even in the absence of infection. And do I have it correct that the cells that are likely to be more resistant have a higher constitutive level of ISGs, or is it the other way around? Because I couldn't get this.
I think what you might be referring to is in figure 5C. So the percentage of ISGs is lower if you have the minor, or what I call bad allele, and that when you're more prone to get the HCV infection.
Right.
So what does that mean? How do I interpret that? What do you mean percentage ISGs? You mean of all the ISGs that could be regulated, there's a fewer?
It's percentage of overall host response dedicated to the antiviral program is therefore lower in the cells that have the bad alleles, that have the minor alleles.
I see. Okay, so generally, I'm saying that the interferon stimulated gene response, the specific antiviral response, is not as good in the guys with the higher infection frequency.
Right.
No.
Right.
Is that right?
Well, the numbers of genes upregulated is highest in the minor alleles or the minor donors. Those are the ones that don't control infection well.
Okay.
So you get a lot of induction because there's a lot of virus there, and that's turning on a lot of genes.
Okay. So that's what I was having trouble with.
So the major donors.
If you had on the surface, it looks like there's a more robust immune response because you're getting more ISGs in the guys that in the minor alleles. But another interpretation is you have more infection.
Yeah. So the minors are having more infection, and they are inducing more genes. The majors are controlling the infection, and as you control it, the number of genes turn on. But there's also a difference in the genes that are induced between the major and the minor. The major seem to induce a certain class of genes which are focused on the virus infection, whereas the minor make a more broadly, and the majors have an effect on cell death. Okay, but the minor have a more broadly focused response that doesn't include this cell death pathways. Okay, so they're having some issues. It's not just the virus that's shaping the response, but the cell. There's something wrong with the host that it's not. It's as if in the major alleles you're getting a finer tuned or more specific response. Exactly.
Right.
Okay.
Right. Yeah. So what they say is the numbers of significantly regulated antiviral genes unique to minor allele donors exceeded that of the major allele donors.
Right.
But when you express it as a fraction of the overall host response, the trend is reversed.
Right.
Okay.
And the cool thing here is that it correlates with the interferon lambda genotype, right?
Right.
So all their gene expression, their interferon lambda, and the virus production or infection, it's all correlating very nicely in this culture system.
I'm imagining some of these authors listening to this, and as I'm struggling to understand it, they're saying, no.
Yeah.
That's right.
Well, of course.
Or we had a 2-week discussion about this.
Yeah, and somebody's thinking, I thought we wrote that really clearly, and we spent so much time on that.
Well,
this is the 4th rewrite.
It is. I mean, of course, they have been thinking about it probably for over a year.
Yeah.
Right? It's tough data to get through because, especially in this,
it's a huge amount of data, and getting it organized and then explaining it correctly is very difficult.
You have to really flip back and forth between the text and the figures, which I don't print anything out anymore, so it's really hard to do.
I do print it out, but they're all on different pages, so it's still hard.
You have to spread them all out, right?
Well, I print double-sided, so that's when I start looking on the screen, and I pull up something on the screen, and I'm looking on the paper, and then, works that way.
I mean, it's not because it's not clearly written, it's just that it's tough data to get through. I hope we're summarizing it.
Right.
Well. Let's see, 2 more points I wanted to make here. They look at individual samples now at individual ISG expression. For example, high infection frequency donors failed to consistently upregulate IFIT1 in the infected and adjacent cells. So they're looking for specific differences. So there are, these high infection donors have a problem with turning on IFIT1. Then there's another gene they talk about, CCL5. It's fairly uniform in infected cells, but intermittent in adjacent cells, a trend not observed in high infection donors. So there's some very big differences between infected and adjacent cells between the high and low frequency donors. So again, implying that there's some response issue. So their conclusions are that ISG expression can be compartmentalized in specific cells, like the infected ones. The uniformity of gene expression varies from donor to donor, and the uniformity of expression correlates with the level of infection frequency. So from person to person, there's a big variation in the response, essentially, that correlates with infection.
Yeah, so this is separating out individual cultures, basically, because previously they had all of the data from one allele type pooled, right? Mm-hmm. And in this case, they're taking individual. They'll have several cultures of different individuals with that allele type, and they're separating them out, and they're seeing sample-to-sample variation, right?
Yes.
But the trend is what they've already reported.
That's right. Yeah, they wanted to make sure that the trend that they had seen was not due to statistical differences that, made things miss. That's sort of what Alan was talking about last year in his statistics article. The last thing they look at is this idea that even the lambda genotype and ethnicity can work together to create an unfavorable treatment prognosis. So, for example, if you have a certain allele of interferon lambda 3 and you're of African descent, you have a bad prognosis. So they had a few donors who fit some of these criteria. 4 donors with the highest infection frequency were either homozygous or heterozygous for one of the clinically unfavorable interferon lambda alleles, and they also had a SNP consistent with a certain African heritage. So it's pretty cool that they could sort through their donors and find that.
Here's a sentence I was looking for about this section about looking at individual cultures or cultures from individual persons is headed stochasticity of the host response. And there's a sentence here that Alan would have loved, but justifying this study. Biologically significant data could be missed in the pursuit of statistical significance.
Right. That's what I was struggling to find before. Mm-hmm. Yes. Mm-hmm. Yeah. Yes, that's the one I remembered. That's exactly what Alan was talking about last week. Yes, thank you. So the conclusion is hepatocytes from donors with interferon lambda minor alleles are more likely to be infected and show a less robust antiviral response compared with cells from major allele donors. Mm-hmm. So this is very interesting because you can imagine now a treatment tailored to individual people, right, who are HCV infected. What's your genotype in these genes that we have found to be important for restricting clearance? Interferon lambda, and what can we do about that, right?
Right.
That's why I asked.
And it also characterizes in more detail sort of the nature of the response to the infection, and I'm sure that will help shape therapies in the future.
It's very cool. There's a point in the discussion they make which I want to bring up, and they say our data stand in stark contrast to those gleaned from the profiling of HCV-infected cancer cell lines, where everything is screwed up.
Right.
And that's a really important point.
They highlight the dangers of extrapolating data derived from commonly used cancer cell lines to initial virus and host interactions in vivo. And you know what I'm thinking? And I agree with them 100%, if this is bad to go from a HeLa cell to polio in people, since when is any animal model going to be relevant? Right? I mean, it's very tough, and this is a good point. Mm-hmm. The other interesting, yes, go ahead.
I'm really impressed by the use of the bioinformatics here. It's becoming, the bioinformatics is becoming more and more important as we move forward. This grouping of genes into related clusters and systems and pathways and stuff is going to get better and better, and it's going to be more and more useful.
Yep. Mm-hmm. They also make an interesting point, which is they say for decades the prevailing dogma was that the chronic HCV infection was driven in part by the ability of the virus to antagonize innate defenses. But they show here that the virus can replicate in cells with competent innate immunity with an antiviral program induced. So the virus doesn't necessarily have to antagonize any innate immunity. Here you have a nice response. You got a lot of innate genes induced, and the virus is still replicating. So it's not as simple as antagonize or not.
So does this complicate issues with regards to vaccine development?
You know, my first reaction to that, Dickson, is that we don't know how vaccines work anyway.
Well, because there is one against hepatitis B. I know it's a very good vaccine. But here's a variety of hepatitis. It's going to be rare and elusive, as they would say, compared to the standard for liver-infecting viruses.
So, Dickson, this is a good point because the innate response is essential for establishing a robust adaptive response, right? So in these individuals, so we can see here that depending on the individual, you know, your genetics play a role in those early responses to infection. So you can imagine that, yeah, these could compromise the adaptive response as well.
So is there a way to boost the good genes that you would like to have respond to protect you in patients that have low response gene profiles? That's
a good question. I mean, I suppose people could
maybe that's what
the
vaccine should be then. Not the virus itself, but one that boosts these innate immune response
So develop
genes.
compounds that boost what you need that's missing in these individuals. Yeah, sure. Yep. I would like to know what Lambda is doing to make it such a crucial player, right?
Is Lambda kind of a new kid on the block? I don't hear much talk of it.
It is relatively new. Yeah, it wasn't discovered. So I have a paper from 2010, which is called A New Addition to an Old Family. It was discovered in 2003. So these are different from the other interferons, type 1 and 2, because their receptors for Lambda are restricted to cells of epithelial origin, whereas the other receptor is on all different cell types. So what is it about the Lambda response that, you know, certain polymorphisms make such a big difference in the hep C outcome? I don't think we know what that is, but these are some clues that they really can influence the response, all the other genes that are turned on. So
it'd be interesting to do this same kind of study with a bunch of different viruses.
Absolutely.
You know,
absolutely.
And I'm sure we'll see that down the road.
All these liver-infecting viruses, right? And they even say malaria here.
They do.
So
they do.
Could be cool, yeah. Anyways, very nice paper.
Yep.
Very nice.
It's amazing that they waited so long to publish this as a single paper. They probably could have gotten 2 or even 3 papers out of this, publishing bits and pieces. I mean, it's a lot of work.
It's a nice story altogether.
But it's a nice, complete study, yeah.
A lot of work. They had the patience to wait.
Okay, time for some email. Hey, Dickson, can you read this week?
I'll try.
All right.
Morgan writes, Dear TWiVers, first, thanks so much for the excellent podcasts. I only started listening to TWiV and to TWIP this past fall. Haven't gotten into TWiM yet. But I'm really enjoying going back through the archives and listening to new shows each week. I'm writing in response to a letter that was read on TWiV 267, where the writer implied that bats and rodents have very large numbers of offspring, perhaps making them better adapted as reservoir hosts for viruses. If I misunderstood, then please disregard the following. However, most bat species only have on average, 1 litter per year, and that litter only contains 1 or perhaps sometimes 2 pups. She gives a reference. While I'm writing, I thought I might also correct a misconception that many people have regarding bats and rodents. While sometimes looking small and mousy, bats are actually more closely related to humans than they are to rodents. And of course, we knew this by looking at the Batman movie.
Morgan didn't say that.
Well, someone had to. And Alan's not here. I live in southern Vermont, just north of Allen. And while I currently work on my master's in environmental studies, my undergraduate work is in the molecular biology. For my master's thesis, I'm studying white nose syndrome in bats here in Vermont. That's a fungal disease. If you haven't heard of white nose syndrome, you should check it out. And TWiV has made me curious about bat immunology. I've started looking for PhD programs in this subject, but there don't seem to be many. Do any of you have suggestions about where I might find a program here in the U.S. for bat immunology? Finally, the weather. It's currently cloudy and 28 degrees Fahrenheit, minus 2 degrees centigrade, with a dew point of 18 degrees Fahrenheit. While it's not currently snowing, we are due for a storm tonight into tomorrow, from which we might get as much as 14 inches of snow. Thanks again, Morgan.
She's got a P.S. here, yeah.
I'm going to read it. P.S. Some of your rodent podcasts, some of your recent podcasts have mentioned fiber arts viruses. I thought I'd mention a related project I'm working on, a quilted periodic table. While I'd love to do a virus quilt, I don't think viruses lend themselves to 2D quilting as well as they do to 3D knitting. So my next quilt might have to be a phage or perhaps DNA packaging.
Cool.
Very nice.
Well, I can tell you that Lin Fa Wang works on bat immunology. He's in Australia. I don't know of anyone in the U.S., but Morgan, I would just email him and maybe he knows someone.
Well, his bats in Australia are much bigger than our bats. They're called flying foxes.
Well, he's the guy who works on these.
Yeah.
So, or it might be that Morgan should just get really good training in immunology and then do a postdoc.
Yeah, that'd probably be the best thing.
Really specialized in bat immunology.
Yeah.
So as I was reading this, I came up with two PLOS pearls. These are these really short, easy to read, sort of micro reviews. One on bats and viruses that talks about why bats might harbor so many viruses and what the issues are, and another on white nose syndrome. Just as background for people who are interested.
Good deal.
Cool. And then she mentions the quilted periodic table, which I think sounds really cool.
It does.
When I was a kid, my dad would bring home calendars from a company called I2R, and once a year they had a special periodic table that was some feature done differently about the periodic table. But I have a quilted virus hanging on the door to my office that my first student made for me. So if Morgan wants, check out my website and follow the map to my lab, and when you get all the way there, you'll see the quilted virus.
You have a picture of it there?
Yeah.
Cool.
On my website, yeah.
For years, I had a periodic table of fruits and nuts.
Oh, neat.
So each fruit or nut began with a specific letter of the periodic table, and so you could read across it. It had all the characteristics underneath there, and it was quite nice.
Kathy, can you take the next one, please?
Sure. Saab writes, hello from Indiana, PA. My name is Savannah Thorpe, and I'm currently enrolled in Dr. Luis Luciano's Biology 466-566 class on Principles of Virology. About a week ago, he directed my attention to TWiV, and I've been enjoying the podcast very much. So first off, thank you for continuing to put it out there for us. I'm certain it's quite a labor of love on your part. Dr. Luciano also recommended that I talk to you about my career goal and see if you have any ideas where I should take them, with regard specifically to virology. I'm an English writing major with minors in applied statistics and biology, as I want to pursue a career in science writing, or translating lab jargon into layman's English to appear in media such as National Geographic, the New York Times, TWiV, etc. I'm a writer by nature, but I find science and math really fascinating and beautiful in a way that scares most of my fellow English majors, who are much more comfortable with poetry or literary criticism. I'm about halfway through my college career, so I figured I should begin gathering data about where I can go with this idea and goal. Dr. Luciano's virology class has exposed me to yet another complex area of study which I'd love to learn and write more about. The idea that's been rolling through my head involves my shadowing or interning at a lab and doing some of its publicity or writing over the summer, but I'm uncertain where to start. Dr. Luciano recommended you guys. If you have any ideas as to who else I can talk to or where I can take this passion of mine, you now have my email, or if you talk about it on your next podcast, I'll be sure to hear it then. Thank you so much for entertaining this email, and I certainly hope we can be in touch. I've very much fallen in love with virology and hope that, even as an English writing major, I can contribute to the field in some way. Take care, Sav. Too bad this is the week that Alan's not here.
Right.
Well, if she wants to intern in a lab, if anyone in the state of Pennsylvania wants to have Sav, shadow the lab this summer, let us know. We can put you in touch. I think that would be
go wash dishes.
Go wash dishes in a lab. If you want to stay in Pennsylvania, there you go.
What was that book? Was it Natural Obsession?
Yeah.
About the Weinberg lab?
Yeah, Natalie Angier, was that it?
I'm looking here. I don't think I have the name right. Oh no, here it is. Here, yes.
Yeah,
Natalie Angier.
Natural
She
Obsessions.
did this.
Yeah, she went to Weinberg's lab and wrote a book about it. Yeah,
right, and basically shadowed them. So Sav might want to, if she's not already familiar with that book, might want to look at that just for kicks as an example of the kind of thing that she's talking about doing. So that was an example of doing just exactly that and having significant success with it. It was good.
I mean, she should figure out where she wants to be over the summer and then write people in that state. And
absolutely.
Say, I'd like to shadow your lab and write about it. I mean, I'd be happy to have someone do that.
Absolutely.
But my lab's too small. You should find a big lab where lots going on.
Right.
So you might remember, at least I do, that I think this is the professor who had his class right in a couple years ago.
That's correct.
And then we had someone come to our graduate program from that college. And it's not too far from Pittsburgh. So depending on, again, where she wants to be over the summer.
Plenty of virologists at the University of Pittsburgh. Mm-hmm. Carolyn Coyne.
That was
Chang and Moore, a bunch of herpes virologists.
All those would be fun labs to shadow. That'd be great.
Yeah, or Jim Pippis.
Jim Pippis. Yeah, that would be good. Sounds like fun.
Mm-hmm.
Yeah.
Okay. Rich, you're next.
Timothy writes, my vote for listener pick of the week, a very scientifically accurate description of the link between vaccines and autism. As somebody with a couple of degrees in immunology and infectious disease, I'm occasionally asked about this topic. Here is the link I'll be sending from now on. And it's a link called http howdovaccinescauseautism.com. And P.S. don't dismiss just based on the name of the link. Don't worry, I'm not a quack. And I did click on this link, and I'm not going to do it again.
Yeah,
it took me to a page that said that that it's been blocked because it's malware.
Oh, really?
At least that's what I got.
No, I don't get malware.
What
do you get anything, Kathy?
I get something with the F word in it.
Yeah, they effing don't.
Really?
Yeah.
It says they effing don't. Yeah, because I thought he was a quack. I said, how vaccines cause autism. But I clicked on it, and it says how they effing don't. That's all that's on the page.
So I got web page blocked.
That's interesting. I wonder if it's your university.
Well, I'm sitting at your computer now, Rich. I could do it if you wanted.
Please don't.
I won't.
Category malware sites.
I would never
It could be
click
that,
on that.
Sites with curses like that are considered to be malware or something. I don't know. Anyway, it's pretty funny. Mm-hmm. I'm glad that it says that, actually.
Well, I'm glad that you guys, found the actual link, because I couldn't get it.
All right. The next one is from Anne, who writes, hello, TWiV team. I am a postdoctoral researcher at UC Davis Vet School, working with Dr. Patty Pesavento of raccoon polyomavirus fame. We had a little exchange with Patty when we did her paper, if you remember. Mm-hmm. Just came across this link to beautiful glass sculptures of viruses. Looks like it was picked up by the New York Times several years ago. Hey, nice.
Good, thanks.
It looks like it was picked up by the Times several years ago, so perhaps this has already made it on the show. Anyways, they are beautiful, and thought I would send in a pic after listening to TWiV for the last year or so. Yeah, we picked it up before the Times. And
right,
was a pick. Actually, someone picked it a long time ago on Twitter.
Yeah, we've had this has come up several times, and actually, I had some pretty significant correspondence with Luke Jerem.
Yes, that's right.
At one point, we were talking about getting a show here or a display of some of his stuff or buying some of his stuff. He's pretty high. He was actually very communicative, and I really appreciated that. But he's also expensive. So,
thanks, Ann. P.S. Currently 48F, 9C, gray and misty, dew point 45 at 11 a.m. I think it's funny. There's an article in The Verge on the glass microbes, right? And the picture they use at the very top is salmonella.
Hello.
Right.
Come on. I mean, at least you put the virus. I know he makes glass sculptures of other things, but why use it to illustrate a virus whose headline is Gorgeous Sculptures of Deadly Viruses, right? Dickson, you are next.
Sir. Michael writes, Dear Vincent and colleagues, I attach a few thoughts on the question sent in by Jeffrey Spelling that you've discussed on podcast 267, in which I included in a blog post, and he lists the blog post from answering something from the deep ponderings on a TWiV question. Hope you find them of interest. I myself am a virologist that emerged from the Glasgow lab in the early 80s and have spent my professional life trying to discover and develop antiviral drugs. Nevertheless, I have only recently discovered and am captivated by TWiV. Keep up the great work highlighting our fascinating role of virology to a broader audience in such an accessible way. Kind regards, Michael.
I think this is so cool that he wrote a blog post on a question that we talked about on TWiV. Mm-hmm. It's called Deep Ponderings on a TWiV Question. It's basically, so the listener had sent in this idea that maybe bats are infected with a lot of viruses because it helps them to kill off other competitors, right?
Right.
That was his idea. So
or that you even might find colonies of bats that had different viromes, or
yes.
no, I guess whatever. Okay, so it's like you're armed with all of these viruses to defend yourself against somebody who wants to invade your space.
Right. So he has some ideas on this, and he talks about them. So I'll put the link in so everybody can go read this. But it's a nice. So it's basically he agrees that this in itself wouldn't be a selection, but it could be a consequence of some other selection force that allows bats to be infected in the first place, right? Because you don't have selection for things that are going to happen in the future. Usually it doesn't work that way.
Right.
So that's pretty cool. We are blogged about. Nice. Kathy, you're next.
So this is Jim writes. Is that right? Is that where I am?
Yeah.
Yeah. Okay. Just in case this hasn't been mentioned as a favorite pick or otherwise, I got into it by way of this link about Robert Hooke's book on micrographia. Click the book cover. But then I got to this page of the site that links to interesting projects on other old books, anatomy, health, biomedical imaging, document conversions, and even nursing homes. Something for everyone. Great place to explore. So this is our friend Jim from Virginia. So I didn't have time to do my homework on all these things.
The first link is really cool.
Yeah, the first link is Robert Hooke's book, yeah.
Oh, and click on the cover. Oh, yeah. Yeah. This is nice. And you can see these pages. So it's been scanned in, and you can see all the images. Oh, this is lovely.
The other page is the National Library of Medicine communications branch, and they have links to all the things they're doing, visualizing rare books, imaging and stuff.
Oh, right.
I have a feeling that Alan mentioned this once, but it's pretty neat. Yeah, it's cool.
Yeah. Thanks, Jim. Good.
Rich, you are next.
All right. Bill writes, Vincent et al. As I steadily catch up on TWiP, TWiV, and TWiM arrears, while steadfastly not missing the fresh honeyed ones, 271, as I exercise my buns off. My mind went to a comment made on the podcast a few weeks ago, to wit, that null research that failed in its objective to prove a thesis of some sort would not easily find a publisher since the volume of papers submitted exceeds what the journals will accept. The nulls being the lowest of the low in this hierarchy, and this would lead to the valuable knowledge that this approach was a failure would not be seen, which would lead to others repeating and failing perhaps more than once to prove this failed approach. Even the ones that succeeded might fail to find a publisher if deemed unworthy. This leads to a waste of mind power and dollars, and it is very much to be avoided. So strong is the need to avoid the waste of resources that I felt certain that some sort of published account should be kept. I suspect the failed research report would be kept in the researcher's own files and in his department, university, etc. as well, but such an account might not be searchable by Google or a search engine, so it might as well be on the moon. Boy, I have a tough time even searching I have trouble searching my own notebooks. I wonder if there is any quantification of the amount of this failed / unpublished research. Would it be 5% or 10% of money spent? no matter if even a lowly 5% is wasted, the avoidance of that 5% waste is the equivalent of a 5% increase in funding, which means literally tens of thousands of jobs across the field. There must be a way that the home lab or home institute could maintain a searchable list in a standardized format of these failed unpublished projects, if only in an abstracted form, so other researchers could readily find them and be able to contact the principals to engage in some sort of dialogue, which would save some money and lead to some new ideas, etc., so that the 5% waste gets closer to zero. I cannot imagine a researcher who would not want all possible data going into a project if it might prevent a serious misstep like a failed thesis or a low grade in the near future. This is so obvious that my mind says it must exist, that even failed or unpublished work must leave some sort of searchable footprint in the data, but your comments on the podcast led me to think that indeed there is not such an archive. If there is not, there should be. How can this gain traction, if indeed it can? Bill. Well, sorry, Bill.
I don't think there is any such thing, right?
No, there isn't.
No.
I mean,
there's no good way to, this is what meetings are for. It's what emails for. It's what the telephone's for. When you're starting a project, if you're sort of hooked in or at any point in a project, this is what you get together with people for and say, have you tried this? Does this work?
Right.
That's the best. That's kind of the best you can do.
Yeah.
Yeah, I was reminded, though, I had to check it out with a couple people and couldn't really find anything for sure, but I know that there was a lot of people who were making transgenic mice and they didn't have any discernible phenotypes, or maybe even knockout mice that didn't have any discernible phenotypes in the ways that they could test them. And I thought that there was a journal where you could publish that or something, and the best I could do from talking to my friend at the Jackson Labs in Maine was that there is the phenome database. So you could, put your mouse there and describe whatever phenotype it had, which evidently wouldn't necessarily be much phenotype. But so, yeah.
The problem with such a database is that it's going to cost money and time to maintain, right? So it's not clear. And, negative results, there's a little bit of mix here that negative is not the same as failed unpublished, right? So some things don't work, and you may view that as failed. But a negative result is important because it tells you what not to do. Again,
and a lot of things don't work because you did something wrong. The pH wasn't exactly the way you thought it was, or the amount of salt that you had in there was different. And you went back and discovered that, and then we did the experiment, and then it did work. So you wouldn't publish the part of it that said, I did this 3 different times, and here's the way I did it, and I screwed up in 2 of those, and the 3rd one actually worked, so I'm publishing everything together. You don't do that, actually. No, right. You just publish the one that does work.
I wonder if we can give Bill any sense of the percentages. I mean, stuff works either because you did it technically right or because conceptually it was the right experiment to do. Only a very small percentage of the time, right?
Richard, imagine yourself as an experimental high-energy physicist trying to create fusion in a tokamak.
Right.
You know how many years they worked with negative results? Many, and they're still working with negative results.
But I think this is one of the reasons that research is so difficult. You have to really want it, because stuff doesn't work most of the time.
Sure, I used to call it re-search.
Yeah.
So the thing that comes to my mind is the series of paper by Fang and Casadeval, right, on reforming science. And they have a number in there about how much research is wrong. All right, so it's different from what Bill's talking about here.
Right.
But that was quite high.
Yeah.
It was almost half, if I remember. But I don't want to call it failed unpublished. What he means here, I think, is negative results, right?
Right.
Yes.
And, I mean, we are starting to see more and more of that published, but it's still a fraction of what's out there, for sure. And but I don't know what % of all results are negative. I have no idea. How many? what about you guys? What % of your results have been negative over the years? Maybe it's a good sampling.
Well, I feel like stuff works. Stuff actually works. I don't know, 10, 20% of the time. That other 80 or 90% is divided, as Dickson suggests, into a couple of categories. One is that I wasn't thinking right; it was the wrong experiment to do, or my hypothesis was totally wrong. And the other, actually, probably larger percentage is because technically I screwed up.
But what fraction is negative? Well, you do the work. You do
the work. You do the right experiment. You don't get the right result. Man, it's got to be at least half, or you don't get an informative result or the result you were looking for.
Yeah, you have a hypothesis. Yeah, and you don't get it. What about you, Kathy? You think it's about half?
Yeah, I would say at least. Yeah, I kind of go with Rich's reasoning.
How about you, Vincent?
I don't know. My experience has been less than half.
You must be smarter than us.
No, I'm not any different, but maybe I view it differently, But
I can give you an anecdote, a very quick one. I was doing some research up at the Trudeau Institute. It was a collaborative effort between my lab and the lab up there, looking at gut-based immunity, basically. And so what we had to do is establish the Trichinella infection there. So I drove up. I stayed with the group for 3 days. I showed them how to handle the infection. And we did everything the exact same way that I did it back in my lab. And they said, great. So they did the experiment. They just wanted to infect rats. And they knew how to lavage orally and everything. And 3 or 4 weeks later, rather, when we examined the rats, they were all negative. Every rat was negative. I saw them isolate the larvae and intubate the rats. So they had to have infected them correctly. What the hell was the difference? So I went back up, and we did it a second time. And this time I stayed up there for a week, and we did 3 or 4 infections. And then I went back to my lab, and 4 weeks later, the same result. So what the hell is going on here? So I finally said, there's only 3 things that can be wrong. The larvae were not infective. The pepsin did something bad to them this time, but not other times. But I brought the pepsin with me to digest the meat to begin with, to get the worms. It's not a complicated process. And you know what the thing that it turned out to be? The tap water. We were mixing the pepsin solution up in tap water. And the tap water in New York State was terrific. But that's New York State water for New York City. It's not New York State water for the Trudeau Institute. They get their water from some completely different place, and it had a lot of sulfur in it. And as the result, it killed the larvae. So something stupid and simple. You wouldn't have published that, but you'd say, from now on, use distilled water. Well, in fact, in all of my experiments, we just switched everything to distilled water and got rid of it like that. But a lot of labs still used just tap water to make the pepsin solution to isolate the worms with. So trivial things, but they can add up to big things after a while.
So I'll revise my estimate, 50%, thinking about all the stuff that my students have brought in here over the years.
Sure,
didn't
of course.
prove anything,
But you're also right, Vincent, it's a matter of perspective. So lots of times an experiment that somebody would say that you could say the experiment didn't work, but the fact is that you get information out of the negative result that reshapes your hypothesis.
Exactly.
So that's right.
That's important because that in fact should be published so that no one else repeats it. Yeah.
Exactly right.
All right. Our last one is from our friend Johnny in Cambridge, who sends us a link to an article which says a Lamborghini Aventador has a brutal frontal collision with a bus in China. And this is for Rich, who made this his pick of the week.
No, it's for Kathy. That's Kathy's Lamborghini.
That's really sad.
Yeah, Kathy doesn't like the way it looks.
Yeah, no, I didn't like the way it looked, so I wasn't going to buy it.
This looks even worse.
Right.
Yeah. Whew.
Right.
All right, let's do some picks of the week. Hey, Dickson, now's your time to start thinking.
No, I have one.
Okay, very good. Let's start with Rich.
Okay. I don't know whether I'm sorry Alan's not here or not.
Oh, this is his stuff.
Right.
Yes.
But I'm not gonna change it because he's not here.
That's fine.
So these are 3 YouTube links that are Alan's little cartoons and verbal essays on what science is. They're called Think Like a Scientist Part 1, Think Like a Scientist Part 2, and Science Controversies. And they're very well done, and just entertaining to look at. I think they're about 5 minutes each, something like that. And it talks about what the scientific method is, how to think about science, and how to apply that in situations where there seems to be a controversy over some sort of issue that involves, a scientist on one side of the issue and a non-scientist on the other side. So, very well done.
Funny.
Bravo,
it's
Alan.
It's funny that you would pick this because one of our letters, which we didn't get to, picked them also.
Oh.
All right. And so I was looking at them because I didn't know they were there, and I went to look at them, and I think they're very cool too. But I do have a bone to pick with Alan about why he's doing this. And he calls this public outreach for real, okay? And I want next time when he's on, we'll discuss this with him. But he wrote a post just before these videos where he said, all this blogging and podcasting doesn't do anything. Let's do some outreach for real. So I want him to really defend that because I don't think what we do here on TWiV is not for real. I think it's real outreach, but I think the videos are great. And he thinks that's real outreach, but that part I don't agree with. Okay, I think they're great on their own, but we're all doing good outreach and blah, blah, blah. Okay.
I think they're reaching different audiences,
Right.
So
Right.
maybe that's...
Well, I think TWiV reaches a very broad audience, which includes the audience of his videos, okay? But he said podcasts don't, and I don't think that's correct.
Yeah, no,
I don't know how he would know that. It's a very interesting issue. He talks about this science ghetto, and we're going to get into this next week when he's back on the show. So it'll be an interesting discussion.
I just want to one more thing. There's a little sort of TWiV inbreeding here. The way I discovered these was I'm Facebook friends with our friend Neva from Buda.
Yes.
And she had a Facebook post on these. So she obviously doesn't have enough to do. She's been reading Alan's blog, okay, and looking at these things. But that's how I found out about it.
That's cool. Mm-hmm. Kathy, what do you have?
Well, I picked an article. It's about the Edmonton, Alberta Worm Invasion Project. And a friend of mine who used to live in Edmonton sent me this article. And it's just full of little factoids that I thought were fun. About 9 years ago, this ecology researcher started looking at worms and found that they're much more common along roadsides and boat launches and not very common in the forest interior, and hypothesized that it was due to human activity. So she wants to enlist a province-wide team of citizen scientists to do that. And in fact, there's an app that you can use, a worm tracker app on iTunes to help with that. But the most amazing fact to me was that, and also to my friend from Edmonton, earthworms are not native to most of Canada. Nearly all of them died out during the last ice age about 10,000 years ago. The worms living in Edmonton now were likely brought over by European settlers in soil with plants and on ships. So, and then I just like the way this particular article is written because they say, earthworms are good, they're the gardener's ally, shredding organic matter. But the squishy compost accelerators have a dirty underbelly. More and more, the voracious eaters are making their way north into Canada's boreal forest, where they chew through leafy layer that covers the forest floor and provides important habitat for myriad organisms. So I just thought it was fun. Oh, and then the other thing on the website, evidently, is that you can drive worms out of the ground using a mustard extraction, a mixture of water and mustard powder. You wait 30 seconds and they just come crawling up.
That's how you get so many to sell.
Yeah, so it just was kind of fun.
That's neat. Very neat. Dickson, you should like that. Oh, you're not a worm fisherman. You're a fly fisherman.
Charles Darwin would be proud. I mean, that was one of his first papers was on the earthworm.
Worms, yeah.
Oh.
Dickson, what do you have?
Well, I have the winners of the Science Envisioning contest for 2013, which was just published in Science Magazine.
Yeah.
I'm a visual person, and some of the images in that one were absolutely stunning, including the cover picture, which was of small I guess they said hair-like projections off of a coral reef, which follow the currents that swirl next to the coral itself and actually help to bring food to the filter-feeding coral. I was knocked out by that. I just have to tell you that to see that picture tells the whole story if you can see the way it works. And then the other one was on the ocean currents that they projected also.
Are you finding this, Vincent?
I am not.
I have the hard copy and I'm looking at it. It's
where is it?
Which
February 7th.
Yeah, it was
February
last week.
7th Science.
Last week.
And what's it titled?
Visualization Challenge.
There we go.
The winners from last year.
Oh, there's a link. Yeah.
Oh, there you go. Yeah, Visualization Challenge.
Quite interesting, and those are quite different from, like, say, 5 years ago, which they were all from microscopy or something that you could associate. Now they're all multi-graphics and bioinformatics driven, and lots of different multimedia just pushed on top of each other to paint the same pictures.
Good.
Quite interesting.
Nice. You like that stuff, don't you, Dickson?
I love it.
Very cool.
I love it.
All right, my pick is a little bit different. I don't usually pick this sort of thing, but it is a television show called Orphan Black. I really don't watch much television, but I don't know, a few months ago, I was listening to another podcast, which was actually recommended by a listener. It's called Security Now. It's about computer security. And Steve Gibson is the host, and he said, you got to watch this show. So Orphan Black is a Canadian sci-fi series. It stars an actress who plays, 5 or 6 identical women who are clones. And there's a bit of science in it, which is interesting. I mean, the point, I think, of the show is that the clones were made by someone who's now trying to get rid of them because they're an experiment that's failed or something. But there's a little bit of science underpinning it. There is actually one of these clones is a graduate student at the University of Minnesota, and she's doing her thesis on Evo-Devo, as she says, evolutionary developmental biology. And she is based on the science consultant for the show, who is a PhD student at the University of Minnesota. And there's lots of talks about DNA and the fact that these clones were barcoded, which is kind of neat. So they can distinguish each other. But, all right, so the science is interesting to a certain extent. But what I find neat is that all these women are played by one actress.
Right, that would be fun.
And they are totally different personalities. I'm amazed that she can do this and keep it straight. And sometimes the two of the sisters or clones are facing each other and talking to each other. Which is, they have to do a split screen, I guess. So it's cool, it's sci-fi, and it's about a science-y thing that's kind of neat. And I love it. I watched all 10 episodes. I bought them on Apple TV, and I watched them, and it's cool. And again, I don't usually watch TV, but I found this cool, and I like science-y related stuff.
Cool, I'll have a look.
Check it out.
Yeah.
And did we have some listener picks?
You got a couple of listener picks here.
We have, did we say any so far?
Did we read?
No.
We seemed like, Jim had a couple of links.
I don't think anyone said pick in the ones we read. I know I had some picks before. Okay, so we had Luke Jerram's glass viruses. Right. Let's read some of these other picks here. We have them here. One's from Steve, who writes, here's a really amazing video. National Geographic has a nice video, as well as a long story by Carl Zimmer about scientists who are trying to learn more about the way the brain works by slicing mice brains into incredibly thin sections, fore to aft, and then using scans of those slices to create what amounts to a wiring diagram.
Connectrome.
Yeah, is this the Obama project?
Well, I wouldn't give him credit for it, but he's certainly behind it.
The goal is to see how all the parts connect and hopefully get a better idea of how they all work together.
Or don't.
Right. Yours don't, Dickson.
Well, I'm looking at the Brain Institute right over here, so I have to agree with the thrust of that pick.
And another from Jacob, who writes, hi, all. In keeping with my apparent bee theme, I'd like to pick this article to share with the TWiVome. Why? Because it's 5,000 bees with sensors attached. Does there need to be any other reason? Also,
We're now at TWiVome. I love this.
We are at TWiVome. Also, as a supplementary pick, now we have supplementary picks. There's a YouTube video linked further down showing a kangaroo giving birth, which is amazing. Possibly not safe for work. I'm not sure. It's a kangaroo. Come on. We're scientists.
Exactly.
Jacob is a molecular biologist in Australia. Cool. All right, that will do it for TWiV 273. You can find us at twiv.tv and also on iTunes. And please go over there if you haven't already and leave a comment or a star rating to help keep us visible because we believe we do real science outreach here and we want to reach as many people as possible. I know that Alan will never listen to this, so I could take digs at him. Send your questions and comments to twiv@twiv.tv. Kathy Spindler is at the University of Michigan. Thank you, Kathy.
Thanks. This was fun. I really enjoyed reading this paper.
It was a good one. Good to have only one paper this time, right? Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Sure thing. Always a good time.
And thank you for setting up Dickson Despommier, who today is at the University of Florida, Gainesville. Thank you, Dickson.
Truly enjoyed it.
Enjoy the rest of your stay.
I will.
He's still got to do his work, you know. He's got to give his talk.
Yeah, he'll be all right. He can do that with his eyes closed. And, you know, Alan Dove was going to text us at the beginning of TWiV from where he is. He's in some warm climate, but he never did. I guess he forgot about us.
Well, he's probably reminded of the joke about clones. Do you know that joke?
Tell us. I don't remember.
It's the guy who clones himself because he can't be in two places at once, so he decides the best way is to clone himself. And there's a slight gene deletion, which creates a Tourette's-like syndrome in the second clone, in his twin. And the twin goes to a party in his stead, and the next thing you know, the twin is swearing and embarrassing himself in public, and they kick him out of the party, basically. And he comes back, and the word gets back to the guy who cloned himself. He says, "You got to stop doing this." He says, "You can't just go out and swear in public. That's not right." He says, "I'm only going to send you to one more event. If you screw up again, I'm going to have to recycle you." So the clone is really frightened, and he goes to this party. He tries to be very good, but of course, Tourette syndrome being what it is, he starts to swear again. And this guy gets word from someone at the party that his clone has gone off again. So he rushes up to the 13th or 14th floor of this party. It was in a penthouse. He confronts his clone. The clone is not remorseful, takes him over to the balcony and pushes him off the side of the balcony and goes back inside. About a half hour later, the doorbell rings. Host opens the door, and there are 3 policemen, and they come in and they arrest this guy. He said, "What the hell did I do wrong?" And he says, "We're arresting you for making an obscene clone fall."
Oh, man, that was so long for that.
It's
Alan,
good, though. You didn't
Alan
mess
could
it up.
probably tell that joke very well. But
you think we're going to be able to clone humans one day?
I hate to say it, I think they're probably trying it already.
Wow. But it won't be permitted in places like the U.S., right?
No, just in North Korea.
Wow. Well, watch Orphan Black. I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.